Hello, my name is Sarah Duhlman and I'm a scientist here at Promega. Here with me today to talk about cannabis research is Mark Bratz. He's a part of our scientific applications group. We put together a series of short podcasts to help you optimize your cannabis research. In this episode, we are going to be focusing on food safety testing. So let's go ahead and get started. So, Mark, why is it important to gender test your cannabis plants?
The nice thing about working with the cannabis food safety market is it's still a food safety market. So the same upfront techniques that you're using for everything else, it's still food. It just has some active compounds in it. So you get the food sample in from whatever you're doing, and then you get it in, you process it. So there's some fun things that you can do with it. Some of the processes that customers have done, you throw it in a bag with some of the medium, and then they have this machine called the stomacher, and all it is, it's literally two paddles that just smash the bag a bunch and break it up. And then that sits overnight. You let the bacteria grow, and then when you get in the morning, it's really simple. You take some of the medium out, spin it down, lyse it with our chemistry, throw it in the cartridge, purified, done. So it's a lot smoother than some of the other workflows where you're getting a plate out and you've got to rinse the food sometimes to make sure you got the bacteria off it, and then you plate the bacteria, and then you let it grow for a couple days, and then you check to make sure it's the correct bacteria, and you go through all that. It's way more simplified to just purify the DNA after you grow overnight and then amp it downstream. So you've got specific primers that you've got that'll target each bacteria's DNA. So you've got a salmonella sample, you've got a listeria sample, you've got E. coli sample, the Shiga toxin, and then you're looking at just whether or not it's there. So it's a lot easier workflow to do than any other of the ones that you are normally done on food safety testing.
What is the time frame from getting your sample to getting results?
So if you're talking about the timing of a protocol, you get after the overnight incubation, after you've mashed it up and let the bacteria grow, you spin it down and lyse. That's about 10-15 minutes, depending on how many samples you're running at a time. If you're running a Maxwell 16, you usually batch in 16. If you've got 48, you got 48 samples at a time. That's when it gets a little bit longer for the upfront part. You're going to have to spin down 48 samples and then lyse them and go through that. It's no more than an hour, I would say, ever. And then once you've got all the samples lysed, you're just throwing it into well one of the cartridge. The cartridge purifies. I think it's like a 45-minute protocol. It's not long at all. Once you've got that purified DNA, you're basically ready to go straight into your qPCR reaction. So you've got your master mix, your primers, and then your sample. You mix them together, amp them, setting up a plate. Depending on how many times you've done it, I would say half a day to 3/4 of a day to run that full protocol. You've already got all of your answers instead of waiting for those couple of days to get the plates going.
What if I have a lot of samples that need to be processed? Is there a high throughput option?
Like we were talking about, you get 16 samples, 48 samples, you're in the Maxwell realm. If a customer wants to go higher throughput than that, we really support customers as they go through the different stages of growth. I know our lab, we have all the different high throughput platforms. We've got the Hamilton, TCAN, Biomech, we've got an EP Motion. We've got all the main players in that high throughput realm. And we also have a field-based team that they go out and they'll actually go to a customer site, make sure they're up and running with the Maxwell chemistries just in bottle format. And then you're looking at just moving, instead of getting all of your samples and putting them in a cartridge, you get the samples ready into a plate. The plate goes on the instrument and the instrument takes care of everything else. So you get out the DNA. And if you have enough automation, or if they're a large enough customer, I know some customers have gone where you get the DNA and then they go straight into that plate, gets moved onto a different instrument, and the PCR is set up on instrument as well. So they basically are just putting the plate with samples onto the robot, and then our team can help them program, get everything going to make sure that it's going to run correctly. So they throw the samples on, and what they get out is the PCR plate ready to go on the amplification machine, the thermocycler.
What are the advantages of using PCR-based method?
PCR can be pretty complex. So even people like me that have used it for years, you still run into issues where you're looking at like different primer sets. You have to design your primers to make sure they're amplifying the DNA correctly, the correct strand of DNA. But one of the real advantages is its specificity. So when you're looking at E. coli, contamination, well, E. coli is everywhere. What we're really worried about with food safety is the Shiga toxin, so the one that will actually make you sick. So the nice thing about PCR is we set up certain primers to look for that Shiga toxin gene. So if there's other E. coli there, it doesn't really matter. What we're going to pick up is whether or not the bacteria that's going to make you sick is there. So when you're looking at different sort of workflows, it's an easier step to just purify your DNA and go straight into making sure that that specific strand of DNA is there. Whereas if you're looking at different plates and all that, I mean, E. coli is going to grow whether or not it has Shiga toxin or not. Then you have to do a secondary test to make sure that it's actually a Shiga toxin containing E. coli. And it's the same with Listeria and Salmonella. There are all different subspecies and stuff like that that may or may not make you sick. What we're looking for is the specific ones that will make you sick. So that's really one of the main benefits of PCR. And when we work with customers, I know I've gone out to a lab where they've had plenty of experience. They've done a lot of PCR. They were running into some troubles with samples that may or may not have been amplifying correctly. They get one that they know should amplify. This has the DNA in it. Why isn't it amplifying correctly? And through optimizing different parts of their workflow, enabling them with the whole food pathogen workflow that we have, and then even just moving to the master mix, the PCR master mix that we have, really help them get past the different inhibitors and stuff that may have come through in their previous workflow. Whereas our purifications generally tend to be really clean. And then on the other side, if you do have anything that does make it through, because these food samples can be pretty dirty, there's no way you can always get everything out. We've had some samples that come through that don't look good. They're colored still, but if you're using the correct master mix, it's going to chew right through it, and you're still going to get the results that you want. So we did a full comparison between like 5 different companies, and the master mix that they ended up choosing that gave them the best result was ours, and that really helped them out.
Thank you for listening today. We hope you've enjoyed this episode of Talk Cannabis. To check out more podcasts and learn more about how you can optimize your cannabis workflows, visit www.promega.com/cannabis.